All the PAs
Is that
know to
recording?
keep me away from
That
Yes.
time is going?
situations.
Yes.
There's one
Good.
PA who makes me go talk to patients who are in difficult situations, like if they're emotional.
You're okay. Can't hurt anybody.
What?
ER and trauma. So I see.
What's wrong?
I see exactly.
What are the things on the side?
You know what I
Yes.
mean?
These?
Yeah. Those are the white.
White and gray,
This is
communicating
what's
green light.
tucking me up because I get really upset.
So they're on different places.
No, you have two sympathetic trunks, right, on either side.
So the gray is the bottom one.
The gray is the top one. It's like just like this picture.
Oh, okay.
Oh, so the white is what we want to find out.
So the white is the indoor, so this is white
The gray is the yellow.
and the gray.
Alright.
Okay. So the scenario we started with downstairs was the initial problem was falling blood pressure. That was the initial problem. There are receptors in the aortic arch that detect the dropping blood pressure. And of course, this information needs to be routed up to the medulla oblongata. Cranial nerve 10 is about as mixed of a nerve as you can get. It has a sensory component, it has a somatic motor component, it also has a visceral motor component. So this is the sensory branch of cranial nerve 10. With falling blood pressure, fewer action potentials are sent. Action potentials are all or none. Like firing a gun. You either do or you don't. The vagus nerve has to send moment-to-moment information up to the medulla. Your blood pressure is constantly fluctuating. But action potentials are all or none. So how does your nervous system code the intensity of a stimulus?
The amount of action potential sent?
The number. So the greater the stimulus, the more action potentials. How do you know the difference between you slicing your finger with scissors versus slicing your finger with a piece of paper?
It's a deeper cut.
It's a deeper cut, and so therefore that's a greater stimulus,
More pain.
and more pain is detected. Both are sending action potentials, but one of them is definitely more painful than the other. And that's because more action potentials are getting up to the brain, to your primary somatosensory cortex, and it's interpreting all of these action potentials arriving in a short amount of time as, ow, that really hurts. So a similar coding frequency is used for blood pressure too. The higher the blood pressure, the more action potentials. The lower the blood pressure, the fewer the action potentials. Either way, it's still cranial nerve 10 that's relaying this information. That does not change.
And it's just a straight shot, Kara, up cranial nerve 10 to the
Yeah.
medulla?
Yeah, it really is. Do you remember when we dissected the common carotid and I peeled away the vagus nerve?
Yeah.
Yeah, it's coming straight off the aortic arch, going straight up that common carotid, following the internal carotid to get back to the medulla oblongata.
But it also goes lower than your
It
heart.
does, but those are the output commands, right? See, cranial nerve 10, I could find branches of it here with the common carotid. I can find it around the heart. I can find it down in the abdominal region, coming and going from the stomach and the intestines too. It definitely is. Downstairs, you were all confused with all these terminal boutons, right? Well, nerves are a bundle of axons, and there's a branch going this way, and a branch going that way, and afferents from here and afferents from there. So we're drawing it as a single neuron, but in actuality, we've got a lot of communication going on here from many different branches within that nerve.
Sorry, real quick. Higher the blood pressure, the fewer the.
No,
No, the more action.
lower the blood pressure, the fewer the action potentials. So the coding matches the stimulus. Greater stimulus, greater number. Lower stimulus, lower number.
Is this the barometric?
Baroreceptor reflex arc.
Why do you say barometric?
Because baro still means pressure. Metric means you're measuring pressure.
Oh, it's barometric. Okay. What do you call it the barometric response?
Like a barometer, what does that measure for us?
Your blood pressure?
Well, our weather pressure.
Oh, never mind.
You use a barometer, you know when it's a high pressure versus a low pressure. All right, so you learned about homeostasis and corrective mechanisms. If our initial problem is a drop in blood pressure, then what should we do to bring it back to set point? Increase blood pressure. So what division?
Sympathetic.
Sympathetic. Now, of course, there are descending tracks that would go through the spinal cord to get down to a level where we would have white rami. Where do we have white rami?
T1 to L2.
Those are the only places where we have our indoor. Where is your heart? Is it above or below your diaphragm?
Above.
Krista, your,
Did it go off?
no, it just is asking for
Signing.
Wi-Fi.
Yeah.
You did it?
She did it.
Yeah.
Thank you.
I can still see me. Okay, so do you think we should send a descending track down to T12, L1?
No.
No. So we're going to target the higher portion of your sympathetic division, right? Maybe T1, T2, because this is definitely where the level of the heart is, right? So a descending track would come down through the spinal cord until it got to a level of the spinal cord where we had the IMLCC, right? T1 through L2. We don't want to go below the diaphragm. Our heart's up here.
Yeah.
Okay. So on your test, I might say you're at the level of T1 through T4. That should tell you you're definitely still in the range for the sympathetic division, but that you're at a level appropriate for the heart. We're not going to the abdomen. So a descending track comes down to the IMLCC. For simplicity's sake, I will not ask you to draw a descending track. But it would exist because, of course, this is a reflex arc. It must be circular. But we won't worry about those descending tracks. I only held you responsible for one descending tract. Just one.
The intestine?
No, the descending tract from chapter 17
Spinolateral.
was for control of your axial muscles. That was the only one I asked you to know. Okay, so then you pick up your reflex arc here. Here we have our preganglionic sympathetic cell body. The preganglionic fiber exits through our exit door. That never changes. It's always the ventral root.
So lateral greyhorn, right?
The lateral great horn, but specifically if we're at this level, it's called the IMLCC. Exits through the ventral root, enters an anterior primary ramus for a short time, enters a white ramus, and this preganglionic fiber will terminate on a postganglionic neuron. Remember, the sympathetic division has short pre's, long posts.
Can you say it one more time? That exact sentence.
The sympathetic has short pre's, long posts.
Thank you.
It's reverse that for the parasympathetic division.
Is
So-
that because it takes longer in the parasympathetic division for your body to calm down?
It's
Is that
not-
why the axons are?
It's not so much a time thing, it's just that developmentally when they were growing,
Okay,
right, that happened when you were an embryo and a fetus. It's just that was their gene expression. The pre's went most of the distance for parasympathetic with very short posts. Jim?
So the sympathetic efferent, the sympathetic motor, they leave out of the lateral, and then the somatic leave out of the anterior. Is that right?
Ventral.
Lauren?
If it's a somatic motor neuron,
Yeah.
the
sure.
cell bodies are down here
Down
in
there.
the ventral gray
Okay,
horn.
all right. And in terms of knowing like the origin and termination, like that nerve fiber sheet that was the supplemental that you had, that was very helpful, but it has like 10 million.
And I said, when it gives you the proper nuclei and ganglia, for example,
Yeah.
I said, don't worry about that.
Okay.
Just stick to a nucleus or a ganglion.
Okay, all right. That's helpful. Thanks.
So now the postganglionic neuron, stop and think. How does the axon want to leave? Are we needing an anterior or posterior primary ramus? Because they are going to lead to body wall effectors. So
It's
we
gonna
don't
bust
need
out the
that.
back.
it's gonna pierce through.
We're going to have it, remember the heart, I drew the heart over here, but it's somewhere in here,
Mhm.
in the thoracic cage, right? So, we don't wanna go out to the body wall, we wanna go deep. So this is gonna break out, and it will terminate in two places on the heart, the atrium and ventricles. And that's why the sympathetic leads to more heart rate, but also force of contraction, and there comes our blood pressure being brought back up.
So what we're not drawing here is the message from your medulla oblongata to your vertebrae, right? Okay, great. And that's the descending tract vision. Great.
Mm-hmm. So if you wanted to draw something here,
Yeah. Could be as simple as that, but it's not as simple as that. It would come in through.
well,
Come through the door
this
for it?
picture is discontinuous with this one,
Okay.
right? Here's my spinal cord, so think of it coming all the way, and this is a transverse section through my spinal cord. No, that's the sympathetic trunk.
This is?
Yeah. So here's your spinal cord. Think of that going all the way up to
Oh.
the medulla.
Okay.
I know it's so
It's so
weird to think
weird.
in 3-dimensional, and our textbook, of course, is 2-dimensional. It's so hard.
So, the brain, cranial nerve 10, is the one who decided to send lower or less action potentials to make blood pressure go down.
No, the initial problem was your blood pressure was down, and the cranial nerve 10 is simply relaying that
That
information.
message to the brain.
Mm-hmm. It's just the messenger. Now, how would your picture change if the initial problem, do we all have this?
Mm-hmm.
I can erase?
Yeah.
Okay. How would this change if our initial problem was an elevated blood pressure? It still is a reflex arc, so always start where the problem is detected. That's always your starting point. Now we have blood pressure going up.
Yeah.
Okay, so that's your starting point. Those are your receptors. What will happen?
Your
It'll
receptors
parasympathetic
go.
Cranial in the
response.
brain,
That's our ultimate outcome.
Okay.
That's the outcome we want. But our initial problem is blood pressure is too high. Jim is correctly saying we ultimately want to lower blood pressure, bring it back down.
Oh,
And he's saying parasympathetic should be our response. And that's true, but we gotta get there.
okay.
So we still have to relay the information first. That's still carried through cranial nerve 10. Cranial nerve 10, when it's a sensory portion of cranial nerve 10, that nerve does not care if the resulting output should be sympathetic or parasympathetic. Its sensory fibers are merely doing their job, saying, don't care what the medulla decides, I'm just giving you the message. This is what's happening down here. So cranial nerve 10, what kind of action potential frequency would it relay?
Hi.
Higher. So there would be a lot of action potentials going up to the medulla. Now the medulla oblongata is the one that gets to say, here's the appropriate output command. Jim correctly said it should be parasympathetic. All right, now you have to think of your nerves to relay down to the heart a parasympathetic message. This, using IMLCC and sympathetic trunk, parasympathetic nerves cannot use sympathetic structures. Period. End of story. They don't share their structures. That's why they are housed in very different places in your body. The output, the nerves are different. So if you get the test version where blood pressure goes up and you're drawing the corrective reflex arc to lower blood pressure, are you using this picture at all?
No.
No, you're not. Because that's, at this level, that's sympathetic. And you all said the resulting of this issue should be parasympathetic. Well, then draw for me parasympathetic reflex output command. Are you down here in the pelvic bowl? No. So do you need to use sacral equivalents to target your heart?
No.
That's ridiculous, right? So no S2, S3, S4. We're not using a spinal cord picture. We're using the cranial equivalents. What are the 4 cranial nerves with a parasympathetic influence? 3, 7, 9, and 10. Does 3 even get down to the throat? No. Does 7? No, 7 targets salivary glands, right? 9, it gets down to the throat, but that's it, the throat. So we need 10.
Again.
We need cranial nerve 10, but we need its visceral motor branch. So, the cell bodies for this sensory branch, they would be in a ganglion just outside of the medulla oblongata. A ganglion. Does anybody have those supplements?
Mm-hmm.
I promise you, I will not ask you the names of the ganglia or the nuclei. But if you were to look in the supplement that says summary of the fiber composition of peripheral nerves, it has peripheral nerves or cranial nerves and spinal nerves. And on the very back sheet it says cranial nerve 10, the vagus. And you'll notice it has afferent, efferent to somatic motor or skeletal muscle, but also visceral motor. So it's listed there. And it says, where do you find the location of the cell bodies for its afferent fibers? Cell bodies for afferent fibers are always, always, always in a ganglion. When it was a spinal nerve, it's the dorsal root ganglion, but for cranial nerves, it still is a ganglion. And they are a collection of cell bodies close to the brainstem. We can actually pick them up with tweezers. We had to dissect these when I was in medical school, and we had to be able to identify each ganglion. I don't even want you to do that, but their names are there, superior and inferior ganglion. So guess which one's on top? Okay, so I drew the ganglion. It's outside the spinal cord. Now look down. Preganglionic efferent. That's visceral motor efferent. Where are the cell bodies for efferent fibers? They're going to be in the ventral gray horn, or in nuclei of the brain, or in the lateral gray horns of the spinal cord. Depends on what kind of efferent fiber we're talking about. Since we're talking about targeting an involuntary effector, we're going to be talking about either inside nuclei in the brainstem or here. But which response did you say we needed to adjust for an increased blood pressure?
Parasympathetic.
So do we use this option?
No.
No. That means we're going to use nuclei in the brainstem. And look, that's listed here. Dorsal motor nucleus of the vagus. I don't care if you know that. It's a nucleus. So I can draw the nucleus in the medulla. That would be a collection of preganglionic neuronal cell bodies inside the central nervous system. Cranial nerve 10, its visceral motor part, comes out of the brain stem, travels down your neck with the common carotid artery. We dissected that out, didn't we? It's going to go real close to the heart, maybe to the epicardium, but then it stops, and what is it going to synapse with? Postganglionic neuronal cell body called a terminal ganglia.
Doesn't parasympathetic have a long
Long pre, short post.
I thought that for parasympathetic had the opposite.
Parasympathetic has long pre, short post. The postganglionic neuronal cell body is in the wall of the effector. Remember terminal ganglia?
So then sympathetic has long pre, short long
Short pre,
short
long post.
No, I didn't.
Which one is the longer word?
Parasympathetic.
That's how I remember it. That one has the long first neuron. Sympathetic is a shorter word. It has the shorter first neuron. Work? Someone's phone.
Is that it?
Do you want more?
Oh, no, I'm just saying,
That's it.
just to clarify too, higher blood pressure, we don't even need to bother with the spinal cord. It's just the brain here.
But if you look at your homework packet, I gave you these options, and I'm going to assess, do you know which pathway you're using?
I'm sorry.
I'm telling you, some of your classmates are going to be drawing both. I see it every time. It's a grading nightmare. Don't expect these grades to be posted quickly because it is a nightmare. And it helps me if you use different colored pens.
So that's what
Yeah.
we do, different colors, but we don't necessarily have to be labeling everything.
I will ask you to label some things too, just like I did
Like in
in your homework packet.
Here, it's gonna be released ACH.
From both, it would be ACH, yes.
So what is the postganglionic? What's that called?
Postganglionic parasympathetic neuron.
That's just what it's called.
What is it called again?
Parasympathetic postganglionic neuron.
So for cranial nerve 10, or that's picking up the receptor that the blood pressure is too high, its cell bodies in the ganglia.
Yep.
But, and then, is it telling the medulla oblongata that activates?
Yeah, so in the
The
medulla
ganglions?
oblongata, if you want to, what you
It's
could
really
do
like
is,
a big deal.
of course, to have integration, we're talking about interneurons.
Right.
So in the medulla oblongata, there's an interneuron that would
Yeah.
connect the two.
Then he tells or he activates
It,
It.
sure, whatever you wanna call it.
the medulla activates the nucleus, which.
Yeah. Look, do you remember in Unit 3, and I told you about nuclei in the medulla oblongata, and you were like, this nuclei thing. I was trying to develop your vocabulary, and you learned about cardioexcellatory center and cardioinhibitory center. Guess what? There you are. There's your cardioinhibitory center. That's what we were talking about.
Right, cardio, for deceleration.
Yeah. Cardioinhibitory, reduce heart rate.
I get it.
This is a clarification question
We
as
have
well.
low blood
For
pressure.
the afferent, for a sympathetic reaction, for an afferent, the vagus, is there gonna be cell bodies in the ganglia also, which you just didn't draw?
Right here.
Yeah, it's the same.
Those are ganglia.
Cool.
Let's see, I drew it outside the
I was
brainstem.
just wondering.
Yeah, it wasn't there when you were doing the sympathetic.
No, I didn't. But you're right.
But
So
it
makes-
is the
yeah,
It's the same.
yeah, yeah.
Because the ganglia is always gonna, I mean, the
Outside.
cell bodies.
Yeah,
are-
I didn't draw that in my first picture. I should have.
Yeah, that's fine.
So we've done.
So what would you ask us to label, like just on the spinal cord?
Tell me what neurotransmitters are released. Put in if you had the test version where you were using a sympathetic output, I might ask you to identify the white and gray radius.
Yeah, except you flip it if you
Oh, yeah.
the gut motility increases.
Can you show me where the central canal is?
Yeah.
If I asked you to put an X on a dorsal root ganglion, could you?
Really, I thought it was the big one.
If there was one.
You
Yeah.
do, but it's just, I mean, it's just, you know, use this thing here.
I thought white. Oh no, white's always lateral. Never mind.
So think of this being straightened out this way. It's weird that the indoor is an overshoot.
Yeah.
Yeah? I know. Anatomy's weird.
Just use one of your
And then
fingers.
so
Just use yours.
if you're asking us to draw a two-neuron chain that would speed up the heart, then we would be doing the
That first
the
picture that I
sympathetic,
drew.
right?
Can we do gut?
Sure.
Yeah,
Gut
Is it increase and
gut.
decrease in gut motility?
Yes.
That's what we're really doing.
Yeah.
So when would you want to increase in gut motility? What are you doing?
Rest
Bar-
and digest.
You're digesting, right? That's parasympathetic control. If you're running for your life, now's not the time to have to go big body. Shut things down.
So
That's it. So we have reflex, arcs.
and aren't they
contact,
both always releasing
that axial thing, and
AC,
that
acetylcholine?
For parasympathetic,
And then for sympathetic, it's acetylcholine and then like epinephrine. Okay. Is that always just for motor? Is that always just okay? This is my sympathetic. So it's acetylcholine being released here.
So the sympathetic is
Or
acetylcholine
here,
but
and
then
or
There is an
here and
exception.
here. So it would just be
There is
here
an
and here.
exception, of course.
Acetylcholine and
For the
then
involuntary
epinephrine.
vectors, it's
What's released
acetylcholine
here?
and acetylcholine.
An interneuron.
No, for
Or,
us,
I don't-
yeah, but no.
Is acetylcholine released at the
From the afferent fiber?
but no, but
Yes.
Yes.
on
I'm
the
confused.
yeah.
This is my sympathetic one I drew.
Oh, I like that.
So this is the one we did right before this one.
Yeah.
So it's like heart rate is
So now
low. You need to
Yeah.
increase heart rate.
Sure.
So you send. This is the
I
vagus
think
nerve,
I
right?
think it's
So is
something
acetylcholine released right there?
Okay, so for
No, I see the
sense,
powder release.
hold
Here.
on,
Only from the.
hold on,
I think.
Hold on.
Okay, she's
Hold
gonna.
on.
She's gonna.
Am I right?
hold
She's
on.
gonna tell.
I know, if you think the
Oh,
difference-
Good for you all, you're starting to get to the more advanced level.
Just give me the whole stack,
Thank goodness.
you. You know, I think for high blood pressure, it's going to be released acetylcholine because more contraction in the heart.
Well, I know that it releases acetylcholine and then epinephrine. I just don't know where.
No, I think that
For this
part is from
No, that one I get.
to the Vegas.
But for
You're talking about the
Still
gut
not
motility
dumb.
or the first one?
Still talking about this.
oh, this is so
I
good.
think we don't have, but I don't know. We don't
I'll
have acetylcholine.
She'll answer.
She'll. I'm
I'll
just gonna
get
wait
ya.
one second.
I'll get you.
I'm okay.
Let me just finish drying my butt cheeks here.
So no lateral grapevine,
I
because we're
am,
down
I
in the
am.
lower. Oh, sorry. Just kidding. Lots of butts. Sorry.
Okay, now, the question that I am hearing is, what neurotransmitter is released with these sensory neurons? Well, don't they want to relay some sort of something happening to you?
Yeah.
So acetylcholine is an excitatory neurotransmitter. If it helps you and you want to put acetylcholine, then put it. Acetylcholine is used in many different places in the nervous system beyond targeting skeletal muscle, beyond this 2-neuron chain. It can be used elsewhere in the brain as an excitatory signal. Why not just put excitatory neurotransmitter? A sensory neuron needs to relay information. You don't want it relaying nothing. So there are other excitatory neurotransmitters. There's glutamate, not just acetylcholine. Glutamate is highly used in the central nervous system. It's enough if you just put a plus. With increasing blood pressure, you'd get more neurotransmitter release, right? You've all said more action potentials. With decreasing blood pressure, you would still get the release of neurotransmitter, just not as much. And that's how the medulla knows how to swing the physiology. It has receptors for this neurotransmitter. If more of the receptors are filled with it, then that tells the medulla there's a lot of action potentials coming at me. If fewer receptors are occupied, then that's how the medulla knows that the vagus nerve decreased its action potential frequency. It's all about receptors and how much they're filled. It's a lot like your gas tank. You have more gas in your tank after you fill it up. When the needle starts to get close to E, what do? You start being more mindful of where you are and where you need to go and where's the most local gas station, right? Don't you? You start to think about, oh no, can't go that far, I need to fill up first. But if that needle is closer to your F, you're full, you're not worried about it as much. That's what your medulla oblongata does with these receptors. The more they're occupied, that's the only way they know there's more signal. Fewer of them, less signal. What does that mean? What do I need to do?
Increase blood pressure.
Right. Okay?
Got it. Thank
Okay. Now, this one that you wanted to do was for gut motility. Now, this one also would be a reflex arc. But I didn't ask you to draw it that way. I just said for you to complete the last half of the reflex arc, where you're telling me the output, increasing gut motility or decreasing. Are there receptors in your gut? You betcha. These receptors are monitoring the acidity of the food you just ate, and if it's disagreeable with your gut, there will be increased gut motility to get it out of you. And you know when that happens, right? You eat something, it hits your intestines, and you're, oh man, that's going to come out real quick. So for this one, I did not ask you to draw the reflex arc, just the last half, the output command. There is, again, a picture of the brain. And in your homework packet, I also gave you a picture of the large intestine the small intestine and the stomach. So large intestine. And do you remember the division line where the vagus nerve cuts out?
The left colic flexure.
So it's right here. Everything else upstream of that is the vagus command. So when you eat food, it goes into your esophagus, to your stomach, to your small intestine, all parts of it. Even though your small intestine dips down low, when it first developed, it was higher up. That's why the vagus could get to it. The ascending colon and transverse. By the time you get to the descending, to the sigmoid, to the rectum, and anything truly in the pelvic bowl, pelvic splanchnic nerves have to give that parasympathetic command. So in your homework packet, I asked you to draw a 2-neuron chain to increase gut motility of your stomach. How would you draw that? Which division has more control? Parasympathetic. Should you be drawing anything then with this picture? No, because this is showing you sympathetic trunk. And we use different freeway systems for these two divisions. So, are we above the left colic flexure?
Yeah.
Yeah, so which nerve are we going to be playing with?
Vagus.
Absolutely, it's the same start. We're going to have the nucleus, the preganglionic cell body. It's going to come out of the brainstem, get close to the stomach, and what is it going to do? Synapse with the terminal ganglia in the wall of the stomach. Which neurotransmitters will these two release?
Acetylcholine.
Both, right?
Mm-hmm.
And you're done.
So this is for increasing
So the
gut
moral of the
motility.
story is get the parasympathetic test.
Exactly.
It's so easy.
Can you label what is this then?
That's the preganglion.
It's cranial nerve 10, which provides the preganglionic neuron, right? And what would you call this?
Postganglionic.
That's the postganglionic cell body, but a collection of them in the body wall or organ wall is a terminal ganglia.
Oh, true.
Cell bodies embedded in the membrane of the effector.
So preganglionic, postganglionic. Cranial nerve 10 is providing the preganglionic neuronal cell body and axon. Bundled up in that cranial nerve 10, when we peeled it away from the carotid artery, if I could peel open that nerve, we would see many axons in it. And some of those axons were afferent, sending sensory information. And some of those axons would be efferent. And some of those, if we could do molecular studies, we would find which ones were visceral versus somatic motor. We can.
So, parasympathetic innervates directly on the organ?
No? You mean the pre innervates directly on the organ?
Right.
No, it has to have a postganglionic partner.
No, that's not what I meant. The parasympathetic uses the turn-on chain to go directly to the effector, whereas the sympathetic has to go through the spinal cord.
The sympathetic will originate in the spinal cord, but
Right.
it will still use a 2-neuron chain to ultimately innervate your effector.
I'm gonna try and say
I'm getting there. Ashley's wheels are, they're starting to gain momentum.
So is there a time when, so if you had an upset stomach, say, and you were sending afferent neurons to the brain, would there be a case where if you had an upset stomach, it might tell the sympathetic system to slow it down, or
No,
Yeah.
so if you've eaten something that's disagreeable,
Mhm.
parasympathetic is going to gain control to increase gut motility.
Oh, link
You're
is
either going to
yeah.
get it out by vomiting or diarrhea,
Okay.
but your gut motility will go up.
And it would just travel up the cranial nerve 10 again.
For the
Oh,
sensory,
yeah.
yes.
And then,
Because it wants it out.
Uh-huh. Cranial nerve 10 provides a lot of sensory information from your gut.
okay.
There are sensory fibers that will tell information about your gut that are also carried through sympathetic routes. But I'm not muddying those waters.
No, I understand.
So, yes, in physiology, you learned about these different reflex arcs from the gut, and if you've eaten something that's bad, you vomit or you have diarrhea. And have you ever noticed that when you see someone vomiting, you want to vomit too?
Oh, yeah, you would.
That's an evolutionary
Yeah.
response. Because chances are we've all eaten
Yeah.
something
Yeah.
bad.
So, with something like IBS, for example, that's from the intestine? Or is
No.
that the stomach overly activating the nerve?
It can be an overactive reflex arc, absolutely. And so those
I have
people,
that.
yeah,
Such a
you eat
bummer.
something and then you have, you
Yeah,
know,
then it just
it's
goes-
nasty.
It is, yeah,
Yeah.
it can be very.
So something is, there are chemoreceptors lining your gut, and certain foods might set you off faster than others, right? Maybe
Right.
with more acidity,
Yeah.
and you have to watch
typically.
that. There you go, your
Or
reflex.
gluten tends to activate it too, so.
So that could be because of an allergic response
Yeah.
causing, again, a haywire with that reflex.
That's interesting.
Sarah,
I'm curious as to where the receptors are in this scenario.
where are they?
Yeah,
They are actually,
because you started in the medulla.
I know, I said you're doing the last half of the reflex arc.
Oh, that's why. But they would be in your stomach,
Yeah.
and it would go from your stomach up, and then it would come back
Yep.
down. Cool.
Yep, so like I was telling Jim, you've got stretch detectors. Have you ever eaten so much where just thinking of putting something else even in your mouth makes you want to throw up? You got stretch receptors, you've got chemoreceptors that are monitoring the chemicals in the food you ate. Absolutely.
Kara?
Yeah.
Very cool.
Can you? I don't know how to word that question.
Well, can we move on to decreased gut motility?
So on the test, we won't have to do the full
So
people,
that's
like from
from
the
No.
door.
Yeah.
The ones for full reflex arc, baroreceptors, which we just
I
did,
don't understand what the cell body.
knee jerk, and cross extensor. Those we drew afferent integration and efferent. For increased or decreased gut
So they're all
motility, I'm just
Here,
going
they're
for the
in
output.
just a
Then
little
why
bit.
did you draw that on the
Yeah.
outside and barrel?
That's all.
Cool.
Because that's where it was in the ganglia, because that nerve has ganglia that is too. So it's either-
It'll
it's
pay
either
off when
going
you get to
through
fizz
a
and
ganglia or
farm.
a neuron. A neuron's in the brain.
In farm, they won't review any of this with you. You better have your terms down with ganglia,
So, like,
nuclei,
see,
pre
look at
and
cranial nerve
post.
10. Do you have that? Oh, we don't have this. So see this here?
Quite frankly, even if you had a question for clarifying, they probably won't remember it.
Yeah.
It's been how many years since
Yeah.
they've had that?
So the cell bodies are in the superior ganglia. Ganglions are always outside.
If you ask them.
So that's where it was
Well,
going,
you get
that's
that
where the
now.
cell
Ask them
bodies
that.
are.
Oh, yeah.
That's what she's like. Well, ask
So,
them.
whereas with this, it's-
Let me look it up.
Or you look it up and Google.
I'm not
Well, I
really
don't
sure.
mind saying I need to look it up,
anytime
but
you
at
do
least I'm
this,
responding to your
Yes, yes.
question.
anytime-
Versus more often than not, they'll say, that was a prereq, you look it up.
How are you supposed to memorize when it's
Or they'll
in
tell you the
nuclear,
wrong
when it's in
information.
nucleus?
Either way, shouldn't you be
Well,
looking it up to
because-
verify?
Yeah.
Oh, yeah.
Okay.
Afferent
But even when we try to verify what we know,
Yeah, it's
sometimes
just
they don't know.
more confusing than.
Because it takes
afferent
time to
are in
actually
ganglion on cranial
verify
cells.
things,
That's
right?
why I have to remember, and then
It's exhausting to answer all your questions.
And some sympathetic
I don't know, it's been a while
This
since.
is here, you know,
What are you talking about?
Is it okay, you can pass it or you don't?
No,
You got
I
the
know.
basic sounds like
Okay, let's do the.
My job by getting you all to draw these and over and over and over, I'm hoping it'll stick. And in Fizz, it'll stick a little bit more. And in Farm, it'll stick a little bit more.
This can't be a hard answer, there's probably just one piece I'm missing. I don't understand why the cell body is in the ganglia outside communicating with the nucleus here for the barrow.
What's the sense of
That was
Why
the,
is it in, for the gut? When heart rate is increased. in the
wait, are we
barrow-
talking gut or baroreceptor?
There.
I'm comparing the two.
Okay,
When the cell body is inside of the ganglia for CNX, or CN10,
yes,
for the baroreceptor, it's the cell body's in the ganglia
Yes.
outside and it's communicating to the nucleus to
yes,
decrease the heart rate.
yes.
The cell body for the gut for increased motility.
I didn't
is-
draw it. I said,
No, no.
it's not a reflex arc I'm asking you to draw. In this example, I'm only asking you to draw the output command.
But it would have been the
Yeah.
ganglia,
Yeah.
because it's the same nerve, right?
Yeah, so there are sensory fibers going up through cranial nerve 10, relaying the acidity of what you just ate.
Okay. So this is just down, so you would have had a
I'm just
separate
asking
chain.
for the output.
So there would have been receptors there to
Yeah.
the ganglion outside of the cell body
Sure.
communicating, so basically
Absolutely.
it's sending this back down. So it's just the second portion.
Yes,
So it's the same, there's no difference. Right?
there isn't. I'm just asking.
So I was asking what is the difference? It's the same.
It's the same.
I just wasn't
A
picturing.
reflex arc has 5 parts. Always 5 parts. In this example, I'm only asking you to draw the last 2 of the 5.
Okay.
The outgoing command, efferent fiber, and effector.
Okay. So increasing gut motility, draw the last 2.
Mhm.
That's what you just did.
Just
Now
the
we're gonna
output
go for
command.
decreasing.
Now we're going to do decreased gut motility.
Cool.
If we want to decrease gut motility, we could start with a reflex arc again. Of course, there are receptors. These receptors would be responding to whatever condition. Maybe someone is taking a drug which impairs gut motility. It doesn't matter. Afferent fibers relaying the information. Output command. All I'm asking you with these gut drawings is output command. That's it. Why? Why am I not asking you to know the receptors? Have you had your
Because I
lecture?
have already listed it in the other questions.
Well, have you had your lecture in the digestive system
No.
yet?
No.
But have you had your lecture on the cardiovascular system?
Yes.
Okay. Just trying to be fair. I think it's abusive already. I'm trying to be fair in my abuse.
Okay, so gut decrease.
Decrease. Now, which division are you playing with?
I'll teach it to you now.
Sympathetic.
Sympathetic, right? So are we gonna draw a cranial nerve?
No.
No, we're gonna use IMLCC, right? Alright, so the preganglionic cell body is in the IMLCC. The preganglionic fiber still exits through a ventral root. The outdoor is always the ventral root. It's going to enter a white ramus and enter the sympathetic trunk. But we want to get to the stomach. Is this above or below your diaphragm? Below, good. Very good. And so, do we want to go back out to the body wall?
No.
No. And because we're below the diaphragm, we are going to break out the back wall. But we're not going to synapse in the ganglion first like we did with our baroreceptor. We got to get closer. We want to get closer to the gut viscera. So it's going to enter a splanchnic nerve and go to your collateral ganglia. You remember the ones that are on your major blood vessels in your abdomen? Celiac trunk, superior mesenteric, inferior mesenteric. In this case, we'd be going to the celiac trunk because that's really close to your stomach, right? So this preganglionic neuron just wants to get closer to its abdominal viscera. It's going to use a collateral ganglion, which is also referred to as prevertebral. You should know by now that there are at least 2 different words for a given structure in anatomy, except for when there are 7. This is an example. And this specific one in our example is the celiac trunk. That's not the only, right? There are 2 others, superior mesenteric and inferior mesenteric.
Where's the splanchnic nerve in
A
that?
splanchnic nerve is
Right
a
there.
nerve that carries preganglionic fibers.
Okay.
Anywhere?
Well, doesn't this carry preganglionic fiber here? But what is this structure called? It's not a splanchnic nerve.
Ventral.
It's a ventral
Ventral
root
root.
because the preganglionic fiber isn't the only kind of fiber you would find there. When it is nothing but preganglionic fibers and that's it, then we call it a splanchnic nerve.
When there's, I'm sorry, say that one more time.
When there is only preganglionic fibers, then we call it a splanchnic nerve. This is not a splanchnic nerve because we find other efferent fibers going out of the spinal cord, right? When we find somatic motor coming out through here. So we're not going to call a ventral root a splanchnic nerve because it has other kinds of motor outgoing fibers,
Okay.
okay? So
I was gonna ask about directly after the celiac trunk, would that be splanchnic nerve?
Yeah, it's
after here,
Yeah,
this one?
right there, yeah,
No, because then what do we have?
because then we have the 3, like,
We have postganglionic,
oh yeah,
yeah,
and that's not a splanchnic.
you're right.
1 plus, we're not going below, are we?
yeah, in this lateral form.
If we're going to the stomach, we're not going to the lower, we're not going to S2.
Well, yeah.
No, but those are parasympathetic anyway.
Right.
Oh.
But, okay,
I was
so
asking,
splanchnic
There's
is only
splanchnic
for-
nerves for both parasympathetic
okay.
and
Oh, right,
sympathetic.
Oh my goodness.
Would we have a pelvic splanchnic nerve on the test?
We can. If I ask you to increase gut motility below the left colic flexure,
Yeah,
then I would have to give you that other picture.
okay, that's
Right.
what I was after, where you can clearly see it.
The ones like this?
Mm-hmm.
Yeah. Like this.
I slept 5 hours and I have not eaten or drinking. And I'm going to go eat and drink before my next class.
Where you saw this picture.
Right.
Mm-hmm.
So here's your dorsal root, here's your ventral root, here's your anterior primary ramus, here's your posterior. If you were asked to increase gut motility below the left colic flexure,
Yeah.
you would then use your sacral lateral gray column. Your preganglionic neuron would be there. It would still exit through a ventral root. It would go through a pelvic splanchnic nerve. Splanchnic means preganglionic fibers only. It would get close to the intestinal wall,
And then the post would go to the wall.
and then it would be in the wall of the effector, right?
Mm-hmm. Okay.
So this is for below colic flexure left. For increase in gut motility, you would need to draw this option. If you're ahead of the left colic flexure, you're using the cranial nerve 10 option. If you're shutting down the gut motility,
Nothing.
sympathetic doesn't care, colic flexure doesn't care, you're still using this option to shut down gut motility.
Oh.
So we just drew the sympathetic response.
Yep, to shut down gut
So
motility.
we're gonna have acetylcholine
Acetylcholine,
release there,
and then here,
epinephrine.
epinephrine.
But we don't want. Okay, is epinephrine's an excitatory? Am I saying the right things?
Well, and here's the lovely part of my answer.
Okay.
What were the receptors I gave you for the sympathetic division?
Nicotinic and muscular.
Receptors.
Oh, receptors.
Nicotinic and
That was for
Alpha.
parasympathetic.
Oh,
Alpha
alpha
1,
Oh,
beta
beta
1,
one,
beta
beta
2,
two.
beta 3.
Uh-huh.
So you see, a neurotransmitter is technically itself not excitatory or inhibitory. Rather, what determines if that neurotransmitter is excitatory or inhibitory depends on the receptor that binds
That
it.
totally makes sense.
So in this case, the receptors here would have a shutdown effect. Yeah.
But you're still releasing
epinephrine.
Great.
This is, those are adrenergic.
They are,
receptors,
you're right.
Is that the right word?
They
And
are,
so
no, that's the right,
specifically-
adrenergic receptors.
But specifically then it's alpha one, beta one,
Yeah.
probably, those are the specific adrenergic.
Yes, so adrenergic means adrenaline, which is another word for epinephrine.
Yeah.
They bind epinephrine,
No.
but the
okay.
response from those receptors
Where it's
binding could
going
either be
different.
inhibitory or
Or
excitatory.
excitatory, and they go to target different parts.
Yes,
with the
you're right.
Shut down gut motility?
Well, it's still releasing epinephrine. It's just depending on the receptors.
No, I'm saying the
Fine.
second one, the
Yeah.
sympathetic one.
The se- that, yeah.
Oh.
Alright, bye guys.
So what if I told you a sphincter controls the exit from the stomach? And what if I told you this epinephrine causes the sphincter to constrict?
Uh-huh.
Then can something pass through it? You've shut down gut motility. So you think of shutting down gut motility as decreasing gut contraction, but you also have to remember sphincters are muscle too. And one way to stop the movement of chyme through your intestines is to close sphincters. Can't get it to move through those doors. So think of it from the perspective of your sphincters. Everything's locked up tight.
I always do.
Sphincters are good muscles.
So, when we're looking at the precentral or the preganglionic, it's going through the white
Ramus.
ramus. Okay.
You're talking about the sympathetic drawing?
Yeah.
Yeah.
Yes, the red.
Yeah, it's going.
Yes.
Okay, that part is the white.
It's
I
still
mean,
a
it's,
white ramus.
okay.
So this picture is showing you many ganglia of the sympathetic trunk: white ramus, gray ramus, white ramus, gray ramus, white ramus, gray ramus. And I've drawn 3 splanchnic nerves, you know, maybe one's going to the celiac trunk, superior mesenteric, inferior mesenteric.
But they said it's not a. I'm sorry.
This would be
Oh. Then why were we talking about that?
that.
Oh yeah, no, the where the red is initially going,
Here,
like right there.
here.
Yeah.
This is a ventral root.
But why? I'm
Can't call it a splanchnic
Right.
because we would have somatic motor neurons with their somatic motor fibers
Right.
coming out to voluntary effectors.
Yeah.
So since they
So
use
a
the same
splanchnic
pathway.
can
We
only
only have right.
create.
Okay. I just was confused why we were even talking about that being. Was there a question that was saying would that be?
Yes.
Oh, okay. Okay. Oh, sorry.
It's okay.
How did they work all this out, the anatomy?
Great question. So there are a series of eloquent studies where they use radioactive signals and they were able to follow, because these are cells, and they have movement of nutrients all along the axon in both ways. So we can give a radioactive signal and follow its movement through an entire neuron, both forwards and backwards, anterograde versus retrograde. So we were able to find nerve endings, insert these radioactive signals, these metabolites, follow how the cell transports these up to the nucleus, and that's how we were able to find the nuclei. If we find a collection of neuronal cell bodies, we can give a chemical signal that's forward moving, anterograde, and follow where it terminates. I remember taking an embryology class in lab, and Dr. French was our instructor. She was amazing. And we were able to design our own experiments in embryology. And I remember one of my classmates wanted to follow a developing germline in an embryo, a chick embryo. And she actually purchased these radioactive materials, and they were able to insert it into these primitive germ cells. And as the chick embryo developed, we could see different parts of the chick embryo with color in it, and that told us where those cells went.
So cool.
I know. So that's how we
That's
figured
great.
out. Yeah, it's pretty amazing, and the technology has certainly changed a lot now. Those early experiments were very time consuming. Nowadays, what we can do is stain a particular cell if we know that, oh, these are filled with this neurotransmitter. Well, then we stain them, and where they are, they light up, and we know that's what kind of neurotransmitters they have. We can even give a different stain where it'll highlight the entire axon. Just stain it instead of relying on this chemical signal to move forwards or backwards throughout the axon. Yeah, it's pretty neat.
I mean, you know what blows my mind is that nothing we have made, like people have made, the most sophisticated machines come even close to how intricate and beautiful and detailed all this is. I mean, it's miraculous when you think about it.
Mm-hmm.
I mean, and I try to when I'm worried about tests and things like that, just take a step back and say, wow, this is like, because it's amazing. I mean.
But to get back to your question, this isn't just some theory of the circuitry. This has been tested.
Yeah, which is great.
Yeah.
I mean.
By watching these chemical signals. So this is not just something that I'm making up and these are my rules for Kara's game of chess. It's not Sheldon chess, you know, where he's got the 4-dimensional chess pieces. It's not made up.
Which is fair. I mean, because when I took psychology,
Mhm.
that all sounded made up to me, and I had a really hard time with
Sure.
that class because it just sounded like a bunch of hooey, you know.
And I agree with you on that one. There's a moment in a traditional psychology class where they do deviate from the hard anatomy, the wiring,
Yeah.
from this part of the brain to this part of the brain. How do we know that? We've inserted these radioactive materials or stained materials and have followed the connections. Magnetic resonance imaging also helps us. So we go from these true relationships to then this, well, here's what they do. And really, we've gained what they do based on injury to people. That's how we've learned what these centers do in the brain, physiologically. And there's a little gray area because then you have to ask people around that individual, how were they before the injury? How are they after? And the waters get muddied because you and I are very different people, and how we perceive reality is different. How we describe what's going on around us is different. So it's hard to find people with the exact same injury, the same lesion size of the brain.
Yeah.
And then you
See
take
that.
your personality differences, and that's where you get your kind of black box.
Yeah.
This is what we think is going on.
Yeah. Yeah, fair enough.
But there really isn't a way to do that legally in humans. I hate to say it, but a lot of what we learned about these centers in the brain also happened during World War II.
I figured. Yeah.
Because the only way where we learned some of these centers and exactly what they do were in lower animals, and we would actually go in and slice at different regions of the pons or medulla and watch the outcome of the animal. Well, rats don't talk. And that's like feeding behavior, sleeping behavior, respiratory behavior. We can quantify those things. But when you're talking about behavioral interactions in speech, there's only one empirical way to test that. And that's to take human subjects and do the same experiments. And that's not legal. But they tried.
Yeah, yeah. That's right.
This might be a repetitive question, but how do we know if it's going to be going to the collateral or prevertebral? Like, do we have to know which one, if it's going specifically to celiac trunk or?
No, just prevertebral.
Okay, so it's always, that was always going to just.
So if
Either
you're
prevertebral.
targeting a sympathetic response below the diaphragm, and you're going to the gut, you're using this option,
And that's always the prevertebral.
prevertebral.
And that's the one that branches into the celiac and the mesenteric.
But we're not going to have to say if it's, okay.
No.
What about for, if it's, are we ever going to be using that for parasympathetic? Well, no, not that, but I'm saying to say which ones it's, no, it's in sympathetic. Oh, never mind. Sorry, my bad.
You're
I
right.
was getting confused in my head.
But that helped a lot.
Yeah, this is amazing.
Thank you so much.
Aren't you glad it wasn't today?